Far below sunlight, a transparent animal pulses through water pressed by kilometers of ocean. Its tissues appear delicate enough to collapse at a touch, yet they function under immense pressure. The apparent contradiction begins with a misleading image of pressure as a crushing hand. Hydrostatic pressure acts from every direction, compressing spaces rather than simply flattening exposed bodies. Gelatinous animals contain abundant water and relatively little gas, leaving fewer compressible cavities. Their soft forms can therefore remain mechanically balanced with the surrounding sea. This does not make them immune, because pressure also alters molecules, membranes, and biochemical reactions. The mystery is how a watery body preserves organization when its chemistry is being pushed. If you descended beside it, external pressure would rise continuously while its outline might change very little. Inside, dissolved fluids transmit that pressure through tissues with no rigid wall resisting alone. The body succeeds through pressure balance, material simplicity, and cellular adaptation acting together. Its fragility at the surface reveals little about its performance in the habitat where it evolved. Our descent follows the same translucent animal from shallow water toward the abyss. At every stage, the environment changes more than the visible shape suggests. Extreme pressure becomes a molecular challenge long before it becomes a cinematic implosion. Every descent of roughly 10 meters adds about another atmosphere of surrounding pressure. Exact values vary with density and location, but the rising pattern is stable. At 1,000 meters, sunlight has effectively vanished, and pressure approaches 100 additional atmospheres. Cold temperatures often accompany depth, changing membrane flexibility and reaction rates alongside pressure. Marine animals usually lack the large air-filled lungs that make rapid human descents dangerous. Many gelatinous zooplankton consist largely of seawater held within thin layers of living tissue. Because liquids compress only slightly, internal and external pressures remain closely matched. This balance prevents the dramatic inward force experienced by a sealed, gas-filled container. A jellyfish bell moves by contracting muscle-like tissue and expelling water from its cavity. The cue is neural activation. The action is bell contraction, and the consequence is forward jet propulsion. Pressure surrounds that motion uniformly, while elastic tissues restore the bell between pulses. Pinafores often move with rows of fused cilia called comb plates rather than muscular jets. Coordinated ciliary beating pushes water backward and carries the animal through its surroundings. The cue is sensory input. The action is altered comb beating, and the consequence is directed swimming. These locomotor systems work in fluid without requiring buoyant gas chambers or heavy supporting skeletons. Gelatinous mesoglea provides volume and shape with comparatively little metabolically expensive tissue. Its material properties depend on water, collagen-like fibers, and other components varying among lineages. Softness can reduce density differences, helping some animals remain suspended with limited energy expenditure. Body plans labeled gelatinous evolved in multiple groups, including cnidarians, ctenophores, and some mollusks. They do not share one universal recipe for pressure tolerance. Verified mechanics explain resistance to gross collapse, while cellular chemistry explains continued life. That distinction moves the investigation from visible form into membranes, proteins, and reaction networks. Under high pressure, molecular volumes and interactions shift, potentially changing protein shape and enzyme activity. Cell membranes can become less fluid as both pressure and cold constrain lipid movement. Deep-living organisms often adjust membrane lipid composition to preserve useful flexibility. More unsaturated fatty acids can help maintain fluidity, though patterns depend on species and environment. The cue is chronic cold and pressure. The action is biochemical remodeling, and the consequence is functional membrane transport. This adaptation operates inside gelatinous and non-gelatinous deep-sea animals alike. Cells may also accumulate small organic molecules that help stabilize proteins under pressure. Trimethylamine N-oxide, commonly abbreviated TMAO, is associated with depth-related protein stabilization in many marine fishes. Evidence from fishes cannot simply be transferred to every jellyfish, ctenophore, or salp. Gelatinous taxa require their own measurements before specific protective compounds are assigned confidently. Pressure can favor certain molecular configurations, while temperature and chemical environment modify the outcome. No single threshold suddenly turns all biological proteins non-functional. Species adapted to shallow water may fail when exposed to pressures tolerated by deep relatives. Conversely, deep specialists can be damaged by decompression during collection. Their collapse at the surface may reflect pressure change, warming, handling, or tissue tearing together. Researchers use pressure-retaining samplers, biochemical assays, imaging, and carefully controlled aquaria to separate these causes. Even these methods reproduce only portions of the animal's natural history. A remotely operated vehicle can observe posture and movement at depth without decompression. Its lights and turbulence may still provoke behavior or injure extremely delicate tissues. Recovered specimens permit detailed chemistry, but decompression can alter the state being measured. Mechanical models clarify pressure balance without proving that a particular cellular pathway is adaptive. The evidence supports a layered explanation. Limited gas prevents collapse, while physiology preserves molecular function. It does not support claiming that gelatinous tissue is inherently unaffected by any pressure. Depth ranges matter because many familiar jellyfish live shallowly and lack abyssal tolerance. Some gelatinous animals occupy great depths, but distribution must be established species by species. Unknown diversity remains substantial, especially among fragile forms damaged by nets. The visible softness is therefore a solution to some mechanical demands, not a complete explanation of survival. As the descent continues, the watery body becomes part of deep ocean energy transport. Gelatinous predators intercept crustaceans, fishes, eggs, and other soft-bodied prey using tentacles or oral structures. The cue is mechanical or chemical contact. The action is prey capture, and the consequence is energy transfer. Cnidarian nematocysts discharge rapidly when appropriate sensory and chemical conditions activate them. Pressure-balanced capsules and tissues must still execute that cellular mechanism in the surrounding cold. Ctenophores use adhesive colloblast cells rather than nematocysts to capture prey. The cue is prey contact, the action is adhesion, and the consequence is retention near feeding surfaces. These behaviors show that softness supports active ecology rather than passive drifting alone. Some gelatinous organisms migrate vertically, crossing large pressure and temperature differences each day. Their tissues experience gradual compression during descent and decompression during ascent. The cue is the daily light cycle. The action is vertical migration, and the consequence is feeding access with changing risk. Tolerance of those cycles may differ from permanent adaptation to a single abyssal depth. Gelatinous bodies also package carbon into fecal material, mucus, carcasses, and prey consumed by larger animals. When this material sinks, it can move surface-derived carbon toward deeper ecosystems. The magnitude varies by region and event, making global estimates uncertain. Soft bodies are undercounted by nets because they tear, pass through meshes, or become unrecognizable. If you judge abundance from preserved fragments alone, their ecological role can appear artificially small. Understanding pressure tolerance also guides the design of samplers that avoid destroying the evidence. The human steak is not copying a mythical incompressible gel, but learning how biology balances water and chemistry. Future studies should observe the same identified animals across natural depth transitions. Pressure-retaining collection can test physiology without imposing an uncontrolled decompression injury. Lipid analyses across related shallow and deep species could reveal membrane adjustments tied to habitat. Protein studies must compare pressure performance at realistic temperatures and chemical conditions. Biomechanical measurements could quantify how mesoglia stiffness and elasticity change under pressure. Genomic patterns may suggest adaptations, but experimental function remains necessary. Long-duration vehicle observations could document feeding, propulsion, and repair in undisturbed individuals. Population surveys need gentle optical methods to include forms that conventional nets erase. Researchers should distinguish permanent deep residents from daily migrants encountering pressure cyclically. They should also separate hydrostatic effects from cold, oxygen supply, food scarcity, and handling stress. No single gelatinous blueprint will explain cnidarians, ctenophores, salps, and mollusks together. You may still see delicacy when the animal's bell or lobes fold around passing water. That flexibility is compatible with strength because strength here means maintaining function, not resisting all deformation. The surrounding sea enters the body's mechanical story without flooding a separate air-filled interior. Cells then carry the harder burden, keeping membranes, enzymes, nerves, and muscles workable. Some molecular details are established broadly, while many taxon-specific solutions remain unmeasured. At abyssal pressure, the transparent swimmer endures through balance and adaptation, never because extreme pressure has ceased to matter.